presentation from Charles Whitfield.
Is that coming through for you all now?
I'm not hearing any volume at the moment. Arunima?
Everyone.
There we go.
Unfortunately, I can't be here today, so this presentation is pre-recorded. If anyone's got any questions, then please feel free to contact me. Air-assisted orchard spraying is easy to get wrong, and I'm sure quite a few people have seen scenes like this with spray drifting across orchards. Good spray application is not about applying as much spray as you can into the orchard area. It's about maximising how much active ingredient you can get onto your target, the trees, and minimising how much spray hits everything else. Fixed rate spray application set up correctly can do a much better job than that you can see in these photos, but it still treats orchards as a uniform target. However, orchards are definitely not uniform, and variable rate spray application has the potential to reduce input costs and improve crop management, leading to better yields and reduced environmental impact. Before we get onto the variable rate spraying, I want to just show how variable orchards are. A well-maintained orchard may appear nice and uniform to the human eye, but it definitely isn't. There is substantial variation in many aspects of the crop, particularly in canopy density. A LiDAR device can be used to measure the canopy structure and density. And in the middle image here, you can see an example of a tree row scanned by LiDAR. This data can then be processed to create a grid of canopy density values, which you can see in the bottom image. Looking at a bird's eye view of this orchard, you can see that there are distinct zones of canopy density across it, with lighter coloration in the northern area, the greeny blue coloration in the middle, and darker coloration in the southern end. And if we zoom in, we can see that there is substantial variation in canopy density on individual trees with a mix of light and dark blocks. So this variation is occurring at different spatial scales. Yield variations can be even larger than canopy density variation, with 3 and even up to 10-fold variations in fruit on neighbouring trees. These histograms show the yield in kilograms on the x-axis and the number of trees with that yield on the y-axis in a Braeburn and a Jazz orchard within a single year. There is a large range in yield between these neighbouring trees, from 5 up to 70 kilograms per tree. Yield variations across years can also vary hugely, for example due to bienniality. And you can see some results showing that in these graphs here. So on the y-axis, we've got the total number of commercially viable apples on individual trees across 2 years on the x-axis. So the lines of the graph show the change in fruit count of an individual tree from 2020 to 2021, and the colour of the lines show the proportional difference of those fruit counts. So the brown lines show that the tree's yield has reduced from 2020 to 21, whilst the cyan and dark green colours show that the yield has increased. And there are 3 graphs showing 3 different areas of the orchard, and we can see that the trend varies for each of those zones. Zone 1 had a large decrease in the number of apples per tree from 2020 to 21. Zone 2 was very mixed, with modest increases and decreases in the number of apples. And then Zone 3 had a substantial increase in the number of apples per tree from 2020 to 21. And all of these counts are from the same orchard and the same variety. Okay, so now I've given some examples and results showing how variable orchards can be. I'm going to return back to precision spraying, and I want to talk about how we can map orchards. So I've already shown some data from LiDAR mapping, which we can use to capture canopy density and structure, and we can map all of that data. You can also use drones. So Outfield have been developing systems for scanning and analysing blossom and fruit per tree, and their system uses affordable and readily available UAVs to scan the whole orchard. The data is then analysed to calculate the position of every individual tree in the orchard. Aerial images of the orchard during the blossom period are recorded using specific UAV flight paths and camera angles. This data is processed and the blossom density for each tree in the orchard is calculated along with its position. This data is highly valuable for orchard management decisions, highlighting issues and for future planning. But we can also use this type of mapped data for variable rate spraying. Okay, so we can detect and map key metrics like canopy density and blossom density from UAVs. But what can we do with this mapped information? Well, we can create spraying prescription maps with tailored doses of products calculated for each individual tree throughout the orchard. The acclaimed software company has developed some fantastic spray machine control software that imports the canopy and blossom density data and turns it into spray prescription maps. These maps can be fed into PWM nozzle controllers on the spray machine so that the nozzle flow rates are adjusted according to the map as the spray machine moves through the orchard. The graphic you can see here shows a visual representation of a prescription map for a single row of trees. Each of the squares in the grid has a value for the nozzle flow rate at that particular point along the tree row. And you can see that each tree and its sections of canopy have different spray values, all calculated according to, in this particular example, canopy density. So the video in this slide here shows some early testing of our prototype precision spray machine, testing out the positioning system, prescription maps, PWM nozzles and the nozzle controllers. The prescription map was set to trigger changes in the nozzle flow rates as they pass specific GPS positions, which you can see on the video marked by the green stakes in the ground. And you can see the spray plume from the nozzles switching on and off as the nozzles pass each of the stakes. Lots more field testing, and in this video machine we undertook lots more field testing, and in this video you can see the spray system being tested on trees in a commercial orchard. The orchard was previously mapped and that data was processed to create a dose adjustment prescription map based on canopy density. And in this video you can see the spray machine is moving along the orchard row, spraying trees and adjusting the spray output according to those dose adjustment values in the map. Nozzles aiming at the top of the tree canopy are switching extensively to account for the leading branches at the top of the tree. And at around 15 seconds into the video, you can see all the nozzles suddenly switch off for a second because there's a missing tree. Here are some results of a trial we did comparing the spray deposition achieved by the prototype precision variable rate spray system on the top graph compared to a conventional spray machine in the bottom graph. The x-axis is the nozzle flow rate settings, and on the y-axis is the deposition on the leaves. The top graph shows there is a strong correlation between the nozzle flow rate settings and the deposition on the leaves, and the variable rate spray system is adjusting the spray output according to the canopy density to deliver a very uniform distribution of spray over the individual trees and across all the trees in a row. It's perfect spraying. The bottom graph, on the other hand, shows there's no correlation between the nozzle output and the deposition. There is a very high variation in spray deposition, and the spraying is very unevenly distributed. The precision variable rate spray system I've shown here today, and other systems like it, have huge potential to improve orchard and tree management. There are a range of orchard metrics that could be measured and mapped and used with a variable rate spray system. Fruit thinning agents should be applied according to fruit load and canopy structure. Plant growth regulators should be applied according to canopy density and growth during the season. Pests and diseases should be sprayed according to canopy density and in the future according to maps of their presence. All these things are possible. The potential benefits of using these kind of precision spray machine systems are huge. Adjusting the spray according to individual tree requirements and avoiding to spray the gaps will reduce the quantity of spray inputs being applied, leading to substantial savings in crop management costs and a reduction in drift and non-target contact. Because the quantities of active ingredient and water in the spray tank are calculated in advance, there will be zero wastage. And bienniality can be managed effectively, and the crop load can be optimised per tree according to its size and canopy density. And by improving the accuracy of chemical thinning, there will be less need for manual thinning, and the percentage of Class 1 fruit can be maximised. So all that remains to say is a huge thank you to all the partners involved in developing this precision variable rate spray machine, and to Innovate UK for funding it, and to you all for listening. So thanks very much. And if anyone's got any questions, as I'm not here, you'll have to email me. Thank you for listening. Bye.
Okay, well, thank you to Charles for preparing that for us late in the day yesterday. I think many of you will have already seen the precision dosing sprayer in action. Those of you that attended Fruit Focus last year will have seen it if you made your way on the farm tour to the Plum Demonstration Centre. It is very impressive, and I think, as you probably noticed there, NP Seymour were heavily involved with that development, and if anybody wants to find out more about that, I'm sure they can go to NP Seymour. Charles has made himself available to answer any questions that you may have on his presentation. If you want to send those to the chat, you can, or you can go directly to Charles. His email address is charles.whitfield@niab.com, and that would be probably the quickest way of getting a response and your question answered. So hopefully that will be of benefit. As I say, apologies from Charles that he's not going to be able to present his fluorometer technology project this morning. So that takes us on to our second speaker, and we've got Richard Colgan, who is known to everybody from these days in the past. Richard has been working pretty much throughout his working life on post-harvest technology. He's well known for all the work that he's done with the post-harvest team, both in his days at East Malling and subsequently at the University of Greenwich, the NRI department. Good morning, Richard.
Morning. Can you see my screen?
We can hear you loud and clear, and we can see your screen. So we're
just having issues trying to.
Are you able to see it yourself? Because
I can see it, but I can't move my slides. So.
Okay, try your mouse, because I sometimes have this problem as well. Try clicking your mouse. There you are.
Okay.
That's fine.
But I can't move back. Can I go backwards? All right, hang on. Sorry. Oh, apologise for this.
That's all right.
So I'll start at the top.
Okay.
Okay, so there. Can you see that?
We can. That's it. That's perfect.
Okay, so today I am going to talk a bit about detection and prediction of internal browning in apple. This is with collaboration with my colleagues Debbie Rees and Karen Thurston at the Produce Quality Centre. So why are we talking about that today? So, well, internal browning is become a big issue some years in some varieties like Braeburn and Bramley, sometimes Cox. But there are different types of browning which sometimes get confused, different types, and they have slightly different causes. So we have things like core flush, which are on the left-hand side, as the name says, it's generally in the core of the fruit and can be anything from a slight pinky color through to a dark brown. They have also issues around CO2 injury, where you tend to get sort of browning, but also cavitation in the fruit, these sort of holes that appear due to high CO2. In Braeburn, we have this other sort of condition we call Braeburn browning disorder, which often sits in the outer cortex of the fruit. It can be slightly, generally around the outer cortex. And there's a condition which we've also called diffuse core browning, which tends to be a precursor to core flush when it does get worse. It's more of a glassy appearance, particularly in Braeburn, which not of any commercial significance at this stage, but as fruits, if it does progress occasionally, it can end up being core flushy. And this is an example of just the core flush in Braeburn and the Braeburn browning disorder together. So what are the causes? So, well, there are a range of causes actually. So sometimes it's due to having a low internal oxygen concentration in the fruit during storage. And this can be down to fruits respiring too much, or the conditions of the fruit in the store atmosphere are too low either. So other issues are high CO2 in the environment. So in cases where you are having CO2 in the storage environment, sometimes a too high of CO2 can cause internal damage, especially as a CO2 injury. But what's really important too is the maturity of the fruit going into store. So high respiration rates of fruit entering store tend to be fruits that end up with low oxygen in the internal cavities. And so it's quite important to understand the fruit maturity at harvest. And we have seen in much earlier work, when we had the days of DPA, that where you had DPA drenching of fruit, the antioxidants actually quenched a lot of the internal stress precursors in fruit, and we got a lot less internal browning, although we don't have any DPA today. But what also interesting is understanding how porous fruit are. That also is how the ability of the fruit to exchange gases, both CO2 and oxygen. And that is also related to a certain extent to fruit density. And these will also change with seasonal factors throughout the year. So this is some early work we did, AHD funded work. We're looking at internal browning of Braeburn. And here we had, I think it was 6 orchards of Braeburn, and we looked at the harvest maturity of that fruit. And what we found was that one of the issues was that if you're just using starch and bricks as a measure of harvest maturity, it didn't always pick up some of the more subtle differences in maturation, fruit maturity, when you start looking at more detail in terms of respiration rates and internal ethylene concentration. Obviously, these are more lab-based measurements, but certainly if you look at the 2 orchards E and F, you can see that when we had a high respiration rate, so orchard F combined with a higher internal ethylene concentration, that fruit went on to develop more core flush, 30% core flush, whereas other orchards which had a similar sort of bricks and starch profile had much less. Albeit that fruit was also slightly larger than the other orchards, but certainly having greater interrogation of the fruit maturity can help you tell you more about what might happen during storage. Again, this is some work, again, when we sort of looked at respiration in more detail over a longer course of at harvest time. So this is Braeburn from 5 orchards in this case, where we monitored respiration rate over a course of about 2 or 3 weeks after harvest. And what we saw was the orchards that sustained a higher respiration rates over that period of time were the orchards where the fruit ended up generating particularly more core flush. It's not totally prescriptive, but we did get some core flush in other orchards that had less respiration rate, but certainly it does tell you that fruit maturity at harvest can have a huge impact on the potential for internal browning. So going back to some really early work I did with like David Johnson and Colin Dover on Bramley, this was looking at understanding internal browning of CO2 injury in Bramley, where we were trying to understand what factors led to internal browning. And what we looked at was, we looked at respiration as one factor. In this case, it wasn't as prescriptive as understanding how porous fruits were. So we came up with a coefficient called resistance coefficient. So the higher the value, the less porous that fruit is. This is based on a number of sort of calculations one has to make based on fruit size and shape. But fruits that were less porous tended to have a greater chance for internal breakdown. So this sort of gives you an idea that it's not just about respiration rate, but how much that fruit is able to exchange gases to the outside atmosphere. So this is actually not my work. This is work done in University of Leuven in Belgium, but it's a published work they published. But they were looking at using some very extensive CT scanning, X-ray CT scanning of different varieties, Braeburn, Kanzi, and Jonagold, where they were looking at the air spaces in the fruit, but not only the air spaces, but how the air spaces were connected. And what you see on the left-hand side is a mapping of the oxygen concentration in different varieties, Kanzi, Jonagold, and Braeburn. And the darker the blue color, the less oxygen is present in that fruit. What you can see, Braeburn has much greater zones of low oxygen than say Jonagold, and Kanzi's somewhere in between. And that's partly due to the number of air spaces, but also the fact of the connectivity of air spaces, with Jonagold having much better connectivity to the outside of the fruit. So we don't have a micro X-ray scanner. So we're doing some other work looking at using another way of measuring porosity, which is using ethane efflux. So this is a technique, a lab-based technique, whereby you introduce ethane into a sealed jar of fruit, and leave it overnight to equilibrate. And then you take the fruit out into a fresh, clean jar and measure how quickly the ethane starts coming back out of the fruit over time. This requires using a GC, so it's a very much a lab-based technique. But we've done this for both Gala and Braeburn. And firstly, there's the two varieties of different scaling because of the difference I'm going to show you in the orchards that we identified. So on the left-hand side with Gala, you can see there is a fruit is fairly not more porous, but it's 6 times more porous than Braeburn is. But we could identify orchards, differences in orchards consignments. However, we didn't see any internal browning in Gala in this case, so it just shows you that there are differences, but not always leading to internal browning. Where Braeburn, the porosity was about 6 times lower than Gala, but we could see differences in the orchards consignments, and the fruits with lower ethane efflux values, the orange and blue lines, tend to have slightly more internal browning than the grey line, fruits and grey line. So this gives you an idea, and we've done some extra work with a commercial company looking at this in more detail, and we've had some really good results really in terms of identifying orchards that went on to develop core flush. So this is work we want to try and extend in the future. But one of the issues we have to do with challenges is to try and take it out of the lab into more of a commercial setting where we're not using GC. So we're hoping to look at other sort of sensors we can use for that purpose. This is some work we did on a SafePod project, looking at dynamic controlled storage. And many of you have had talks on SafePod before. This is a unit that fits into a commercial store. I can measure respiration rates of the fruit during storage, and we can use that for low oxygen storage, DCA storage. So this is the work that my colleague Debbie Rees was looking at, some commercial outturn work of orchards. And one year, I think around 2017, we found there was a pretty bad case in some orchards of senescent breakdown in Gala, which is quite rare. But we found that the orchard A on top left-hand side of the screen, that this orchard had quite a high level of senescent breakdown. And it also was the orchard that had the highest level of respiration rate measured during storage. And then we went on to look at other orchards in that year. We had safe pods in the stores, and we ranked them on in terms of how much senescent breakdown was present in those orchards. And then looked at the respiration rate. And there's a general trend, although it's not perfect, that the high respiration rates were present in fruits that had some amount of senescent breakdown. And so this is something which we're quite interested in exploring further, and we do monitor stores where they are having respiration rates are using safe pods and being measured, and it has been used as a way of sort of ranking stores as to which store should be open first if the respiration rate starts to rise. And on the bottom left-hand graph, you can see you have a U-shaped like curve respiration rates that start off high during storage, the beginning of storage, and then they bottom out around December, January, and then start to rise slightly in March, April, where fruits start to become slightly more senescent, I suppose. And those fruits that have a high respiration rate at that point tend to be the fruit that we need to move quicker. So this is the last bit of this talk. I want to talk about that work we're doing with the University of Manchester, using a technique called non-destructive assessment of fruit quality using what's called magnetic induction spectroscopy. This is a group that do a lot of work on sensors. We've been working with them over the last 3 or 4 years on both apple and avocado. So the idea of this technique is, can we determine, without having to cut open the fruit, what is the internal quality? And we've done some work looking at Braeburn and core flush, but we've also had work with other colleagues at NRI on avocado, and also recent work on potato. So we're looking at a technique although it's called magnetic induction spectroscopy, is actually looking at the electrical conductance across produce and how does that then relate to product quality. So I'm not an engineer, but certainly what colleagues at Manchester say is that cells in tissue act like a capacitor. When a cell's intact, they have less resistance. When the cells become damaged, when you put electric current through them, the resistance changes. So it can tell differences in the tissue integrity by how much electrical conductance passes through the tissue. And it depends on the frequency of that electrical input. So what we're talking about here is what's called beta distribution, which is around 10 to 100 megahertz of electrical frequency. And this has been used at that frequency. You can tell differences in cell membranes and then any fluid leaking out of those membranes, which can be indicative of tissue breakdown. At higher wavelengths, and microwave gamma dispersion wavelengths, you can also determine things like water content, which might be useful for dry matter determination. And then using scanning across several wavelengths, you might be able to understand not only about damage, but also potential for ripening and maturation. So this is a prototype they had some years ago with a little conveyor belt passing through this big magnetic coil, you can see. And then they're able to distinguish between fruits that of different damage. So what we've been doing with them, which was some work we did last year, was taking Braeburn fruit, which had different propensities for internal damage. The orchard H21 was an orchard that was good fruit quality. And orchard BP was one that was CO2 stressed and had a highlight of CO2 core flush. And looking at this big graph on the left, you can see that there's difference in the conductance of those tissue from those orchards and apples, which related to the amount of damage going on. So if you look at the graph on the right-hand side, you can see that the BP and the H21 orchards were quite different in terms of their electrical conductance. And that was indicative of the amount of damage that we saw internally. Equally, this is not, obviously, we have done work on avocado, but equally, this is also useful for ripening, this technique. In this case, we're looking at changes in the ripening of avocado, where fruit started off with a day 0 with a particular conductivity, and that level of conductivity increased as the fruits ripened. And so this has great potential for avocado, determining how avocados are, but also potentially maybe for understanding apple maturation too. So I think we are early days of this project with them. It's all very much underfunded work we're doing on our own backs, but certainly some interesting results. So conclusion, certainly the rate of internal browning in apple does increase with the advancement of fruit harvest maturity. Respiration rates at harvest do give a good idea of what you might incur during storage. And also during storage, measuring harvest maturity measurements during storage can also give you an idea of what that store fruit might be doing in terms of potential for damage. But fruit porosity, again, understanding how porous fruits are can really give you an idea of the greater risks of internal damage, particularly for core flush in Braeburn and also in Bramley. And the electrical impedance technology has potential to identify core flush during grading, but probably a lot more as well. Just one last slide to acknowledge, well, lots of people have been involved in various projects, which I'm talking about today, but these are just some of the people that have helped us out over our project. So thank you.
Thank you very much, Richard. Really interesting, actually, particularly for me, because as many people know, I spent some years in my previous life working as an agronomist doing store management and store monitoring work. And interesting about the porosity, because I remember doing quite a lot of Jonagold sampling. I don't suppose there's an awful lot of Jonagold left these days to monitor and store, but I never ever saw any internal browning in Jonagold. So it was interesting to see your results there and how they compared to things like Braeburn and Gala, and presumably that was one of the factors behind the reason there wasn't any internal browning was because of its porosity. Yeah, I think that's true. The work the Belgian scientists did show that that Jonagold being much more porous than any other varieties, so obviously less issues with internal browning. Yeah, I suppose the next question would be where, or what stage are we at with that? Is there any, in terms of sensor development, are we anywhere close to having a prototype or something that the industry can use?
Well, I know the Belgians are trying to commercialize something they're doing on porosity, and we've done some work to convince ourselves. I think the ethane efflux model does work to show us differences in identifying orchards that are less porous and have more greater damage. There are some sensors out there which we can use that are used in other industries for measuring ethane, but we need to do the work to really calibrate them, to take them out into more of an industry setting. But that's work we hope to do in the future, but if we can get some funding. I've applied for some university funding to do a pilot scale study this year. Waiting to hear.
Okay, that's useful. And presumably Cox, which we see less of these days, but presumably Cox is more in the Braeburn category, with a slightly more dense fruit, would you say?
Well, I think Cox has its own issues. I think it's not as dense as Braeburn, certainly, but I think there are other issues around maturity, respiration rates that might lead to core flush in that variety.
Okay, and as regards the respiration rates, again, like porosity, is there anything close to being available to use there, or are we some years away from that?
The safe pod system can be used at harvest time, or many versions of that can be used at harvest time to measure respiration rates, and that some growers do have that system in place. But also during storage, we can use the safe pod system to monitor fruit quality throughout the season. So there are options. Our growers are using it, who have the safe pod system, are using it to measure respiration rates, and so yeah. So it's already commercial.
And potentially in the foreseeable future, it may be an aid that agronomists and store management experts will be able to use as an aid to their work that they're doing, rather than a replacement to chopping apples and looking at vast numbers. Yeah,
I think you keep chopping apples as much as you can. But yeah, certainly an extra sort of interrogation that you have, just to give you a heads up early on that things might be changing in your store.
Good. Okay. Just double checking the chat. Yes, we've got a question here from Jonathan. Is porosity related to the flesh, lenticels, or the combination of the two?
It's mainly the flesh, and it's also skin thickness, certainly to a certain extent. Lenticels numbers, I don't think they've really looked at lenticel numbers, but obviously that's the final exit point of gas. But certainly lenticel numbers would be something to look at in more detail. But certainly, general the air spaces in fruit and how they're connected is really important. You can have air spaces that are not connected, and then you end up with more damage. So the connectivity is as important as the number of air spaces.
Good. Thank you, Richard. I don't think we've got any more questions. If anybody does think of any more, if you do put them in the chat function, I can pose them to Richard later on. But for now, Richard, thank you so much for your time and your presentation this morning. We will now move on. As I said earlier, sadly, our next speaker, Charles Whitfield, is unable to present today on his fluorometer technology. So we're going to move on to the next speaker, is my colleague from NIAB, Mark Helts, who is known to you all very well, who's presented at these events many times in the past with his crop science and production systems team at East Malling. On this particular occasion, Mark's going to be talking about the Plum Demonstration Centre. Good morning, Mark.
Good morning, Scott. Good morning, everyone.
Let me just, before we kick off with Mark, let me just say a little bit about the background to the Plum Demonstration Centre, if I may. I'm going to turn the clock back 12 months. Soon after I joined NIAB, Mark and I convened a meeting of the plum industry to discuss the future of the Plum Demonstration Centre. Some of you will recall that the Plum Demonstration Centre at East Malling came about following an Innovate UK project, which involved both industry partners and research partners. And the purpose of that was to try and raise the quality of plums and, if you like, improve the quality and spread of season of plums so that it became a more profitable crop, because it's always been seen as the Cinderella crop of the tree fruit industry in recent years. That particular IUK project required some demonstration work, and the team at East Malling planted up and established a new plum orchard for that purpose, and it included combinations of rootstocks and tree architecture and various other things that we were looking at. AHDB then took on the funding of that because the IUK project finished and the AHDB Tree Fruit panel decided they wanted to support it. So they funded it for 3 years. And then with the demise of the Tree Fruit Levy and the AHDB Horticulture Group, it needed new funding. So Mark and I convened a meeting, and we got huge response from all the principal plum producers in the UK, not just in the southeast, but in the Midlands as well. And also some of the marketing groups, and they all got together and kindly agreed to put some money into this, so they pay for the maintenance of the Plum Demonstration Centre. So without our funders, we wouldn't have the centre at all. So huge thanks to all of them. And since we had that meeting back last year, we've had a number of consortium meetings with the funders who have discussed what their major requirements are, what they want to use the Plum Centre for. They are focused on some things more than others. And I think a year on, we've made huge progress. And Mark is going to tell us a little bit about what we've been doing and what their high priorities are. So hopefully that gives you the background we need, Mark.
Excellent. Thank you, Scott. Can you hear me?
We can, yeah,
Well,
everything's
good.
fine. Yeah, we can see you as well.
Very good. So yes, good morning, everyone, and thanks for this opportunity to talk about some of the work we're doing at the Plum Demonstration Centre. And thanks, Scott, for the background information to the centre. So if you go to the next slide, please. So this is just a plot plan of the Plum Demonstration Centre at NIAB East Malling. Hopefully many of you will have visited as a Fruit Focus last year, or in, as Scott mentioned, in some of the previous projects. I think we'll also be presenting at this year's Fruit Focus. So if you haven't been, do come along, and obviously you can see what we're doing out there in the orchard. But just to summarise then, I've split the plot plan into two. So here on the road there, this is towards the south of the planting, and then the bit on the left, on the right there, sorry, is towards the north of the planting. So today I'll mostly be talking about some of the work we've done in the area there you can see on the left with the training systems trial. I'll also mention some work we did in the polytunnels, and a brief mention of some of the work we've done in the green area there on the right-hand side, but mostly I'll be talking about the training systems. And those sort of red and blue ovals represent where we've installed data loggers and sensors and rain gauges. And of course, what we're trying to do then is to use that data to inform decision making in the orchard. And so I'll come on to explain a little bit more about how we're doing that in the next slide. And so these are the research priorities of the Plum Demonstration Centre Consortium. And I think it's fair to say the focus is very much around trying to optimise consistency of yields and quality in the main commercial varieties, but also bearing in mind that that needs to be achieved with an efficient and responsible use of water and fertilisers. And so the key areas are then that the Plum Demonstration Centre Consortium wish to pursue are around precision irrigation, precision fertigation, trying to better understand how to match demand with supply, both in terms of water and in terms of nutrients, how we might start to impose techniques such as regulated deficit irrigation that will allow us to use, I think, resources even more efficiently, and may also impact positively on fruit quality and sort of shelf life potential, as well then as some of the effects on some of the ground management practices that we see in many orchards. And so there's an example last year, my colleague Michelle Founding had a project where she was looking at the effects of wildflower strips on sort of beneficial organisms and ecosystem services. And so we were just keen to start to gather some pump priming data around the resource requirement for that crop as well. But essentially the focus is on targeted irrigation and fertigation to really try to optimize yields and quality. Okay, next slide, please. So first things first, before we could do that, the orchard was in some need of attention, because you'll probably remember back in 2021, we had a very late frost. Obviously, we lost all the fruit from that very late frost event. And so the trees were quite out of balance by the end of that year. And so the first job really was to try to use a variety of different sort of techniques to try and bring the the vigor back into control. And you can see there, the first photograph on the left is an example of a root pruning treatment that we applied. And you can see there the guys on the right-hand side who came to visit us, I think probably in May, and this is very much an industry-led consortium. So it was great to see the industry turning up on a Saturday morning to help make sure that we prune the orchard in a way that was appropriate for commercial production. So that was really the first challenge in last season, was to try to bring the different growing systems back into balance following that frost event in 2021. And you can see there some of the, practices that we were carrying out there. Okay, so if you go to the next slide, please. So, as I said, I'll mainly talk about the variety trial, but obviously you'll have seen from the plot plan, we do have some trees in a tunneled area. And so that was part of the Innovate UK project that Scott mentioned, in order to try to help to bring those trees back into control in terms of vigor. We removed all of the skins last year and just tried to apply a dilute fertigation recipe to try and bring them back into control. And you'll see there from the plot plan that those are mostly Victoria and 2 of the peat varieties. And again, we were also wanting to encourage some of the newly planted trees in those areas. Some of the rows were incomplete, and so we have since planted trees on the ends of those rows. And again, then for those trees, we're wanting to try to increase root growth, for example, by applying monoammonium phosphate and a bespoke establishment mix that was provided by Yara. Okay, so next slide, please. Okay, and the next one. So one of the first pieces of work we wanted to do was really trying to understand how to better match demand with supply in terms of irrigation and fertigation. And so we've used an approach that we've used many times before in lots of different crops, and this is a sensor-based approach. And you can see there in the photograph on the right-hand side, we have 2 different types of sensor. That one there where the cursor is pointing to is a measure of volumetric moisture content. So that basically measures how much water is in the soil per meter cubed. And you'll see there the one in the bottom of the photograph then with the disc is a measure of the soil matrix potential. And I'll come on to explain the benefits of using a combination of those 2 different sensors in a moment. But essentially then we place those sensors at different depths within the rooting zone. So at 15, 30 and 45 centimeters, we put the Koya soil moisture content sensor at 45 centimeters in order to be able to quantify the amount of irrigation that was percolating past the main rooting zone. And again, we applied a specific fertigation program that was devised by Yara. And initially then we irrigated when the average soil matrix potential reached a value of -60 kilopascals. And you can see there, as we go through the season, we gradually reduced the irrigation set points. So we were irrigating at different points. I'll explain a little bit more about that in the next slides. And then, of course, we were wanting to see what effects this fertigation strategy had on fruit expansion, yields, and also, of course, fruit quality. Okay, next slide, please. So a little bit about soil matrix potential. So as I said in the earlier slide, measures of volumetric soil moisture content or Koya moisture content or substrate moisture content are quite common. The issue or the challenge there is that those values are affected by the bulk density of the soil. And so depending on the soil type, or for example, at the age of Koya, if we're in soft root systems, those values can vary significantly based upon the age of the substrate or the bulk density. And so we're always very interested in using a measure of soil matrix potential. And essentially the matrix potential is a measure of how tightly the water is bound to the soil particles. And so it's a measure of how hard the plants have to suck in order to try to extract water from the soil. And you can see there on the right-hand side, there are a variety of different sort of sensor systems we can use to measure matrix potential. And I'll explain a little bit more about those as we go on. But just to reiterate the point here then, that the matrix potential is independent of any effects of bulk density. And so I'll explain that point in the next slide. Thank you. So on the graph on the left-hand side, then you have soil matrix potential on the y-axis and soil water content on the x-axis. And you have there, you can see the different lines. They're representing different soil types with different bulk densities. And essentially what this graph is showing is if we look at the wilting point, for example, if we look at the wilting point in each of those different soils, the wilting point varies from perhaps 5% in the sandy soil over to the green clay at about 35%, 40%. So the point there is that the wilting point occurs at very different volumetric moisture contents. Whereas if you look at the corresponding value on the soil matrix potential axis, they actually occur at a very similar soil matrix potential because of this independence of soil matrix potential from bulk density. And so the graphs on the left, on the right, illustrate this point very nicely. This is actually a crop of lettuce grown in 2 different soil types, as you can see. And this is the point at which the soil drying started to initiate stomatal closure. You can see that by the red dot in the middle there. And the point here is that although the point at which that occurred is very different in terms of moisture content, you'll see there on the x-axis, it actually occurred at a very similar matrix potential in those 2 very different soils. And this just sort of re-emphasizes the point then that matrix potential is quite a useful measure to us because it's independent of bulk density. And so it can be used across many different soil types and many different substrate types. So that's why we tend to use a combination of soil matrix potential and also volumetric moisture content. Okay, next slide, please. And so this is how we schedule irrigation over the season. And you'll see there on the x-axis, then we have the date through the season, and on the y-axis there, then we have the soil matrix potential. And for example, field capacity is the top black dashed line, and that's always around -5, -6 kilopascals. And as the soil begins to dry and the water becomes less available, the matrix potential decreases, so it gets more negative. And so on the x-axis there, then as we go down the x-axis from -5 down to -150, -200, that's an indication that the soil is becoming drier and the water availability is becoming more limited. So you can see there the bars in the graph then represent the rainfall events measured at the orchard there. You can see obviously then reflected in that very dry June and July, a very good summer we had, very little rainfall. And so you can see there then initially we started to schedule irrigation when the average matrix potential in the soil in the rooting zone was around 60 kilopascals. And we are applying these events manually at this time by turning on the solenoid valves. When we're imposing these sorts of irrigation systems, we always decrease the step points gradually, so we're not placing the trees under any shock. And so from 60 kilopascals there, then we lowered the irrigation set point to -80 kilopascals. Finally, then before trying to maintain the soil matrix potential, the average soil matrix potential in the rooting zone between -100 and -140 kilopascals. You can see that we managed it fairly well, apart from 3 occasions there when the matrix potential drops very quickly. And that's often when you have a heavy crop load, a very sunny, breezy day with a very high vapor pressure deficit, the soil matrix potential can change very quickly. But essentially then we managed to schedule irrigation based around the data we were receiving from those sensors there that you can see on the right-hand side. Okay, next slide, please. And this is just to say, oops, sorry, no, go back up one. This was just to say then that the graph on the left is the average matrix potential across the rooting zone. You can see there the 3 graphs on the right-hand side then are at different soil depths. And so we've got a very clear understanding of how the trees are using the water in different horizons of the soil in order to inform those decisions. Okay, thank you. Next slide. So these were some of the training systems, or some of the results from the training systems, and you can see the training systems there in the graph. And we have a narrow A-frame, narrow tabletop, an oblique system, and a super spindle. Now, these particular training systems and rootstocks aren't set out in a sort of a randomized, statistically rigorous design, and so we can't allocate any significant differences to those yields. But you can see there in the narrow A frame and also the narrow tabletop, we're getting yields of around sort of 14.5 kilograms per tree, which equates to about a 32-35 tons per hectare-ish. You can see then lower yields from the super spindle system, and again the oblique system then coming in at around 4 kilograms per tree. So very different cropping potentials on those trees. As I say, difficult to conclude any more than that because of the lack of statistical replication. But it just goes to show that we can reduce or schedule irrigation and fertigation quite effectively using those pro systems and still achieve a good commercial yield from those trees. Okay, next slide, please. So, of course, what we want to try to do, of course, is to schedule irrigation based around the likelihood of any rainfall events. And so if we're approaching the irrigation set point on a Tuesday, for example, we wouldn't want to schedule an irrigation event on the Wednesday if we were due to receive a good downpour. And so we want to try and schedule that irrigation scheduling with what's available from rainfall events. And so what we're keen to do then is to understand how we can schedule irrigation in line with weather probability forecasts, which will give us a fairly accurate regional picture of the likelihood of rain and also the depth of rainfall that we begin to see in some of the major plum growing areas. And so that's an area of research that we want to sort of develop, and we're sort of will apply to various funding calls to try to do that. But you can see here then in the table, then we have the water applied in terms of litres per tree, and then we have the rainfall that was estimated over the cropping season, and then we have what we call the water productivity value. And so the water productivity value then is a measure of how productively we're using each litre of water in terms of class 1 or marketable fruit. And essentially the lower the water productivity value, it means the more productive use of water. And so that's something we're aiming to better understand. We're working with the grower partners in the consortium to understand what their water productivity values are, and that will help us highlight best and better practice. And it will also help us highlight opportunities to improve resource use efficiency, both in terms of water and fertilizer. So next slide, please. So mentioned very briefly at the beginning about the trees in the tunneled area. So they are the two, P variety, P619, Elizabeth that was sort of released last year at Fruit Focus, and also Victoria. And you can see they're fairly young trees, and so we were just starting to see the first crop coming off those trees. So around 3 kilograms per tree for Elizabeth, low yields for P619, and about 5 kilograms a tree for Victoria. So again, the main aim there is to try to bring those trees back into balance for the current cropping season. And you can see there just some data on the Brix values that we saw, and also the firmness as well. Okay, next slide, please. So when we come back to efficient use of water, of course, one of the concerns of many growers of stone fruit is not to try to let the soil dry out too much immediately before harvest, because the risk is then that if there's a sudden significant rain event, then we might start to see some cracking of the fruit, as we often see in sweet cherry. And so, and also the plum growers are concerned about that too. And so they're often quite reluctant to let the soil dry down immediately before harvest in case of these rain events. Now, obviously, the weather probability work that we're planning to do will help to assuage some of those concerns. But we also wanted just to test the hypothesis about whether we could dry the trees down significantly before harvest, and then apply a sudden irrigation event in order to see if we could initiate any splitting or cracking in the fruits. And so you can see there then the graph in the blue line there, that's the commercial control, where we're irrigating to keep the moisture content at a reasonable level. And you can see then the matrix potential shoots down in the red line there to quite significantly low values. And then that's immediately before harvest, and then we apply an irrigation event to bring the moisture content back up to field capacity very quickly. And of course, an irrigation event isn't the same as a heavy rainfall event. But despite that, despite this significant drying and sudden irrigation, we saw no effects of that treatment on splits, either in the control or in the soil drying treatments. And so clearly it's something we're interested to do more work in to see whether that result is repeatable over the season and in different varieties. But it does question then perhaps the need to maintain the soil at field capacity immediately prior to harvest. But obviously more work is needed in order to be able to advise growers on how best to maintain moisture content before and during harvest. Okay, next slide, please. And this is really just an observation. And so from the plot plan I mentioned, we were doing some work with a colleague, Michelle, looking at sort of the effects, the beneficial effects of wildflower sort of strips in the alleyways there. And you can see that in the photograph on the left-hand side. Now, coincidentally, perhaps those Victoria trees were very, so they're younger than the trees in the trading area. They had a lower crop load, but they were very, very thirsty, and we applied a lot more water to those trees, and they also started to develop nutrient deficiencies compared to some of the more mature trees in the training systems. And so, of course, we're interested to understand why that might be. And of course, one area we want to investigate further is whether those wildflower strips are competing for essential resources in the soil, such as water and fertilizer. And so we place some sensors under trees with and without the wildflower strips, and we got some indication that the wildflower strips were competing for resources, but nothing conclusive. And I think we need to do more work with Michelle and her team in order to better understand how those sort of beneficial wildflower strips impact on the availability of water and fertilizer. One thing we did see in the soil down at that end of the orchard, in the sort of the north east section, was then the soil organic matter was very low compared to the other areas, and the soil potassium concentrations were very low. And so I think partly the driver behind this a significant demand for water and fertilizers then is a very low organic soil matter content, and that's something we're keen to understand how to improve in the future. And so I think that's just everything I wanted to say. Just a brief update of the work we've done in the Plum Demonstration Centre. As I say, the main focus was to try and bring the orchard back into balance, and we're very grateful to our consortium members in helping us to do that. And they visit several times a year and offer advice and help on how to do that. And so I'd like to thank them especially for supporting this work. As Scott mentioned, they've all been very, very generous with their time and inputs as well. And finally, thank you all for listening, and I'd welcome any questions if there are any.
Thank you, Mark. I'm not seeing any just yet. I have a couple. First of all, and I may have missed it, but you were talking about water productivity earlier on, and you were showing the differences between some of the training systems. And there seemed to be a large difference between the oblique spindle and the spindle. Was there any reason attributed to that difference?
So the issue there, Scott, is that all those at the moment, all of those training systems are on the same irrigation line. So they receive a similar amount of water. And if the yield then is lower in some of those trees, that means that there's a less productive use of that water. So I think the main driver behind those differences in water productivity then was the lower yields on the oblique trees compared to the other trees. But that's something we're wanting to look at this year, is how we can better target irrigation to the different plantings within the orchard by establishing different zones of irrigation using different solenoids. And so we can be far more targeted in how we apply irrigation and fertigation to those different systems.
Okay, a couple of more questions come in. Actually, one of them from is asking pretty much the same question. Another question here: Mark, would you recommend similar matrix potentials for apples and cherries? I know you've done work on this before.
Yes, very much so. And so we've done a lot of work with AHDB funder work and HTC funder work on apple and cherries. And some of those sensors I showed, there was one with like a traffic light system. So very easy sensor to use and install. It's a visual indication of the soil matrix potential. And so you could put that in a representative tree in the orchard and just monitor that to make sure that perhaps you weren't irrigating when the soil matrix potential was still quite high. So I think having those low cost, simple devices would be very, very helpful. And I think the main benefit would be to prevent over irrigating the crops when the soil matrix potential is high enough and the crops don't need it. And so, yes, that would be useful information for stone fruit growers, but also for apple and pear growers as well.
Okay. Just final clarification, coming back to the same question about oblique spindle. The question wasn't so much about the water use, but why was the fruit yield so much lower for the oblique system? Do we think we've got a reason for that?
Well, I think we don't know, honestly. I think at the moment, I mean, there's a very significant difference in yield. So 4 kilograms a tree compared to 14 kilograms a tree. So no, I don't think at this point we know what the reason for that is, but it's something obviously we'll be looking more into in the coming seasons to try to better understand why we have that wide-ranging cropping potential on those different systems.
Okay. Mark, thank you so much. I think we've got to move on, and thank you for sharing. I think this is all very good progress that we've made in the last year, and the plans for next year are exciting too. So thank you for all that you're doing, and thank you for sharing all the information with us today.
Thank you.
Okay, thank you, Mark. So that takes us through to our final presentation of this morning, and we move to my colleague Robert Saville from the NIAB's Growing Kent Medway team. I work very closely with Robert. He's an Innovation Growth Manager. For those of you that haven't heard of Growing Kent in Medway, it is an innovation growth cluster which is supported from the government's Strength in Places Fund. It's been doing a vast number of different projects over the last 18 months since it was set up. NIAB is the lead partner, research partner within this. And Robert's going to tell us a little bit more about some of the exciting things that have been going on. Robert, good morning, or good afternoon, I should say now.
Good afternoon, Scott. Thank you very much. And hello all. So I'm grateful for this opportunity to present on the Growing Kent Medway cluster today and how we are supporting sustainable horticultural innovation. I will start with a couple of slides to introduce what Growing Kent Medway is for those who don't know or have forgotten. And then I'll present some of the activity we have been delivering over the past 12 months to help horticultural food and drink businesses to become more sustainable. So Growing Kent Medway is a 5-year programme funded by Strength in Places Fund, as Scott mentioned. And Strength in Places is a UKRI programme that helps areas of the UK to build on existing strengths in research and innovation to deliver benefits to their local economy. In the case of Growing Kent Medway, we are building on the region's existing strengths in horticultural and plant-based food and drink research. And although our programme is focused in Kent and Medway, we do want to become a sort of place where UK or international people will look to serve this research area. The programme is led by NIAB, as Scott mentioned, and we have also teamed up with the University of Greenwich and Kent, together with Locating Kent and other business stakeholders, to create a nucleus of our research and enterprise support cluster. The cluster has a focus on boosting regional economic growth by delivering and uplifting Kent and Medway's economy through inclusive and innovation-led growth. And we are supporting technology-driven horticulture, fresh produce packaging, food and drink processing, and its supply chains, all with a focus on supporting sustainable growth. Around the cluster's nucleus, Growing Kent and Medway is creating a business network to enhance local collaborations involving research and innovation. So what does Growing Kent and Medway offer? We have various activities which all have a focus on business innovation and supporting the Growing Kent and Medway cluster. So our R&D grant funding programme we're using to support research in our target sectors, and I'll speak a bit more about that in our in some upcoming slides. We offer business support through the Growing Kent and Medway's Food Accelerator, a mentoring programme run by University of Greenwich and University of Kent, respectively. At the centre of what we do is Growing Kent and Medway's at the centre of what we do at Growing Kent and Medway is creating a business and research network and facilitating connections and collaborations through events. The programme has also enabled each of the research organisations involved in the Growing Kent and Medway cluster to invest in new research infrastructure to support our target sectors. We're also conducting a skills assessment, which will identify what skills our sector will need by 2030, and working with colleges and training providers in the region and beyond to ensure that appropriate training is delivered. And underlying all of what Kent and Medway does, all those activities, is our inclusive growth agenda. So Kent and Medway has some of the widest social inequalities in the UK, which is a barrier to achieving prosperity and well-being. And we are driving an inclusive wealth creation so that untapped human capital can contribute to and share the region's prosperity. Okay, so for the rest of the presentation, I will talk you through some of the examples of how we at Growing Kent and Medway are supporting sustainable horticultural innovation. Starting with our research and development grant funding programme, I'll move on to explain how a project we delivered last year called Growing Green has supported sustainable innovation. And then I'll finish on some examples of our networking and events activity. So a range of Growing Kent and Medway grant funding opportunities have been developed to fund innovative projects at all stages of development and are available to companies of all sizes and stages, mainly within Kent and Medway, but there are ways that other companies can from outside of Kent and Medway can engage with this grant funding programme. Sustainability and net zero are at the core of all of our funding competitions. Since the programme started, we have had 2 rounds of funding for large collaborative research and development projects. And last year we ran our first round of business innovation vouchers. These give opportunity for Kent-based businesses to get subsidised access to the expertise and facilities at Kent-based research facilities. And we are soon to launch a new grant scheme which will fund businesses directly to undertake innovative, sustainable projects, either alone or in collaboration. So look out for further information on these later this week. Here's some of the large collaborative R&D grants that were funded in round 1. They started in May sort of time last year. So we have projects ranging from those focusing on increasing production, reducing waste, enhancing nutritional quality, and on carbon sequestration. And later on, or the session after lunch, Matt will talk about work he's been doing on apple canker, which may well touch upon some of the Growing Kent and Medway supported project on that topic. You can find out more about the range of the projects we supported in round 1 on our Meet the Grant Winners page on our website, where you can find a short description of each of the projects, the partners involved, and a short video from the business lead. And at the end of last year, the second round was concluded for this competition, and the winning projects from that round will be announced in the coming months. So I'm now going to play a video to show you some of the projects funded through the Business Innovation Vouchers round one. I've got a little warning there about sound. I don't think I can control the sound, and the music gets a bit bassy in the middle, so have the volume control at hand in case it gets a bit loud for you. So it's just a 1 minute video, which I'll try and play now. Sorry. Okay, I did warn you it would get a bit loud. So just to say, a new round of the business innovations will be launched in 2023. So if that video has whetted your appetite for accessing some of the research facilities available within the cluster, do let me know, and we can start to talk about how we might develop project proposals for that you to receive subsidized access to those facilities. Okay, moving on. So another way Growing Kent Medway is supporting our target sectors to be more sustainable is through the Growing Green programme. So the Growing Green programme was funded through the Community Renewal Fund. It was led by NIAB, with a number of partners, including Low Carbon Kent, which is Kent County Council's sustainability team. It was a pilot project to support horticultural, food and drinks businesses towards net zero. And to achieve that, we gave the businesses involved one-to-one support. We provided training and showcasing. We helped them develop a decarbonisation action plan. And we provided them with a small grant to undertake a decarbonisation project. And this pilot project helped us to, and our partners and funders, to learn what aspects of reaching net zero our target sectors need support with. And it helped us to test and develop methods to deliver that support. My colleague has told me that this bar's in the way, so I do apologise, but I don't know how to get rid of it. But behind that, it says the range of businesses we have worked with on this programme are listed in this slide. I mean, there was some 38 businesses that we supported. There's just some examples of those in this slide. drawn from across Kent, and in the middle we have the variety of different horticultural businesses on the left and food and drink businesses on the right. We produced a number of video case studies to showcase what the businesses that we supported did with their in their innovation projects. And I'm just going to play you one here from Giles Cannon at GFJIB Cannon and Sons. So bear with me.
My name is Giles Cannon. I'm the managing partner at JIB Cannon & Son of Rothway Farm, Plaxtal. We're a family-run fruit farm. We grow apples, cherries, plums and raspberries, and also are reasonably famous for our Kent cob nuts as one of the few growers of that item still in the country. So the project that we're doing with Growing Green is involving cold storage. Cold stores use a lot of electricity during the season, and we have around about 16 or so on site, and we also contract to store elsewhere. And with the recent increases in electricity cost, it's become even more relevant. And then of course there's the green angle and the fact it's better for the planet to use less power. So we looked at a project that would in some way start to chip away at the amount of electricity that we use in our cold stores. And we chose a store that we use virtually all year round, 12 months a year, for various reasons. It stores stone fruit during the summer, and we store raspberry canes in there in the winter. That therefore means any changes we make will have a benefit almost every month of the year. So we chose this particular store at Rothway Farm, and it involves changing the fans to a new sort of fan called NIAB fan, and also adding something called inverters onto the compressor. And these are estimated to deliver a power saving over the course of the season of around about 20%. It's relatively new technology, and we need to see that it works. And if it does work, and both here and no doubt on other businesses who will try this out as well, we can then roll it out to other cold stores. And if you start to save 20% of your power on all the cold stores, then that is a significant number in terms of saving on kilowatts of electricity.
I was muted there. Thanks for that, Giles. So this is part of a series of videos produced as part of the project to showcase how businesses in our target sectors are innovating to help them become more sustainable. And we've got a number of other videos in this series from, for instance, a vertical farm business in Swale, finding an innovative way to keep his produce cool whilst he's out distributing. We helped Nim's Fruit Crisps in Sittingbourne illuminate their food waste from her operations. We helped an Ashford-based distiller to close the loop on otherwise wasted resources, that was water and heat. We helped a Tunbridge-based cob nut processor to efficiently sort his waste streams for which he could find new markets for. And we also helped a Wye-based kombucha brewer to trial a bottle return scheme to reduce the packaging waste from his operations. And we helped a number of other businesses besides. And this series is just to help to inspire others in the industry to see what they can reduce or make more sustainable in their business. So if you would like to view any of these video case studies, Growing Kent Medway has a YouTube channel accessible through our website or through this link. So how can you get involved? Although the pilot of Growing Green has finished, Growing Green will continue to support businesses towards net zero through our Growing Kent Medway project. So this includes the inspiration and showcasing piece that we, for instance, those videos and knowledge exchange. With regards to the knowledge exchange, we are developing a resource hub tailored to our sector to support you in becoming more sustainable. We have an events program and business master classes which feature sustainability topics, among other aspects. We also have the R&D funding program, which, as I said, has sustainability and circularity at its core. And we also, as part of Growing Green pilot, we developed a sustainability audit and decarbonisation action planning process, which we are now we hope to offer in the near future through the Growing Kent Medway mentorship programme. So that's Growing Green. I'm now going to present a few slides about our networking and events programme. And how that is supporting our target sectors to become more sustainable. So the sustainable packaging is a special focus topic for Growing Kent Medway. And last autumn, the University of Kent hosted the inaugural Growing Kent Medway Sustainable Packaging Forum. The event brought together businesses and academics from around Kent and featured inspiring presentations from leading sustainable food and drink packaging businesses. And the event saw the launch of a regional business network to address packaging challenges and targets from the roots up. So that database is being built currently for people and organisations who are involved or interested in sustainable packaging in Kent Medway and beyond. So if you would like to be included in this networking database, there's a short online form which I can share in the chat and after the presentation, or just contact one of the Growing Kent Medway Innovation Growth Managers, that includes myself, by email. So basically the form will ask you what your interest in packaging is, whether you have particular challenges in your business, or whether your business can offer solutions to others. This database will then provide a networking tool to help people find one another, develop project consortia, and also we as IGMs, Innovation Growth Managers, can then help match you up and find funding to bring your projects to reality. With regards to our events programme, we run a number of events focused on various aspects, including sustainability. I have a couple of examples here from the end of last year of tours, events that we ran showcasing net zero initiatives in the region. And we have plans to run more of these this coming season. We also have a whole series of business innovation masterclasses, providing business support for horticultural food and drink businesses. They're open to all across the UK. They're run online, and they can cover sustainability topics among other topics. So I've now got my last video of this presentation. It's to show an event that we held at the end of last year with one of Growing Kent Medway's industry partners, Smurfit Kappa. And the event was designed to inspire horticultural food and drink businesses to consider how they could make their packaging more sustainable.
Where we have recycling and we provide that material to the paper mill, and then that's made into packaging. So we're basically trying to get that closed loop, so we get the packaging customers' waste back into recycling, back into the paper mill, back into their packaging.
So currently we're at the stage where we're sort of trying to develop a prototype package for Indian savouries. But it's been very useful today just looking at the processes, and it's given me some ideas of how to sort of eliminate plastic fully and try and protect the product as well.
We made the decision 3 years ago to go single-use plastic free. We kind of worked backwards and we're sort of finding solutions, and coming here today was really interesting just to see the sort of the raw end of how materials are recycled. And on a commercial context, people wanted a kind of a greener experience. That's what we want to work with Growing Green and develop over the next few years, hopefully.
It's sustainability, it's carbon footprint, it's going to save them money in the long term and save the planet, so it's good all round.
Okay, and once again, that video is one in a series of videos showcasing net zero innovation, and these are also available on our YouTube channel, so which is linked here. Okay, last couple of slides. I just wanted to finish with sort of if any of this has whetted your appetite. I'd just like to sort of leave you and ask you to think about any waste or byproducts or sustainability issues in your business or industry, and equally any inputs which you could potentially source from other industries to replace or substitute less sustainable inputs. Because going forward, Growing Kent Medway are looking to support any circularity opportunities that may be present in your business or industry, and we want to support your business to deliver those and help you collaborate with others in the industry. So please get in touch if you have any ideas. As I say, there'll be opportunities upcoming to help you develop them. And how to get in touch. So there's many different ways to do that. You can visit our website, you can email myself or any of the team, you can follow us on the various social media channels. But the best way to keep in touch of all the opportunities, events, networking opportunities and such that are upcoming is to become a Growing Kent Medway member. It's free. Just head to our website, hit the pink tile in the top right hand corner, which will open up a form for you to complete. Okay, thank you for listening, and back to you, Scott.
Thank you, Robert. I always like the use of video clips. It tells 1,000 words, and it's really helpful to include some of that. I think I would just add to what Robert's saying, that he really has just scratched the surface of what Growing Kent Medway has been doing over the last 12 to 18 months